we'll start with, I guess, basic introductions. So it's a really great honor to be here on stage with you today. We're all in the cell and gene therapy field, but you have truly revolutionized the way medicines are made, either through discovery, technology, or manufacturing. Perhaps to set the scene and tell people why we're even all here, Geoffrey, I'll point to you. You talk often about how we're finally living in a time that technology is allowing biology to become engineerable. Could you tell us a bit more about that, and how does this apply to cell and gene therapy fields?
Sure. First of all, it's awesome to be here. And Dina's taking me back to roughly 20 years ago. So I was an undergraduate focused on chemical engineering. And I feel really lucky to have fallen in love with biology. It was not an instant romance in that I jumped into a lab, was given the most menial, most boring set of assignments, was on the verge of quitting 3 months later, and my advisor had a number of philosophical conversations with me, the essence of which was that, look, everything becomes engineerable at some point, and biology's next. And what I was doing in lab, although we were using the word engineering, was anything but. And if you define engineering to mean we can predictively succeed in doing things, then every time that's happened to a field, chemistry, electrical systems, the whole world has changed. So any chunks of life doing that are going to be a really big deal. And in fact, chemistry and electricity didn't have the advantage of starting with two multi-trillion-dollar industries of food and all of healthcare. So biology has sort of a running start. And in a sense, the categories that we're talking about here are among the best examples of what that transition is going to mean. In that, almost by definition, engineered cells as medicines and genetic medicines are going to do something that has been very scarce and rare in medicine, which is either cure a disease or prevent disease from ever happening. And so if we take them kind of one at a time, genetic medicine means medicines that act at the level of DNA. And the way biology flows is that DNA is the code of life. Nature, billions of years ago, chose DNA to store all of its information. It stores information more densely than any human-invented technology. And it drives the biology of every single cell in our bodies every day of our lives. So you could sort of think about medicine having spent the last 100 years climbing up to the level of DNA. And now that we're there with the first cures taking place, if you're trying to cure a disease or prevent disease from ever happening, DNA is going to be the rightful home for a really big chunk of those medicines. Engineered cells as medicines are also showing the first examples of cures. And if you just imagine kind of piecing apart your biology into all of these wondrous different cell types that allow you to see light, the photons flowing in through the lights in this room, allow your liver to metabolize all of the various chemistries that it's exposed to, allow you to think, allow you to breathe, allow you to allow your heart to beat. In many diseases, in fact, most of the ones that are going to affect our loved ones, a critical cell type is either missing a set of instructions or has lost the ability to function. And the ability to be able to take cells from outside the body, in fact, at times, derive from another person's cells and hide them in your cells while they carry out a critical function, is going to allow us to cure or reverse a huge number of diseases in the coming years. So, as we go from engineering and biology, meaning we can change stuff, to meaning we get to predict what's going to happen and succeed when we set out to do it, these categories are going to be extraordinary.
Thank you. That's an excellent introduction. And I think perhaps instead of going directly into the future, because this revolution is happening right now and is perhaps not being televised, let's talk about the near past, okay? Could you perhaps summarize and tell us, and let's start with you, Jeff, like what have you achieved in the past few years that has truly changed clinical practice and is benefiting patients right now. And you're a perfect example with Spark, which is one of the first gene therapies brought to the market. Rahul, you can tell us more about resilience. And then Geoffrey, perhaps what you're doing with Tessera and Sana with gene therapy and cell engineering respectively. Jeff, why don't we start with you?
Yeah, so basically, can you hear me?
Yeah, can we hear?
Is this working? All right, I'll switch to the microphone. So yeah, the basic concept, playing off of Geoffrey's great introduction there, is that in some cases what we think we can do to try to address diseases is to add back or add in the correct instructions at the DNA level that can help a cell then sort of carry out the biological process that we would consider healthy or normal. And so one of the first ways we thought about how to do that about a decade, or frankly multiple decades ago at least, was the original thought, was to use viruses and actually engineer those as a delivery tool to deliver, instead of the DNA that's inside a virus that might cause disease, to take that part out and put inside, in place of it, a normal copy or the normal instructions that would otherwise allow the cell to have the normal biological functioning. And so that led to, frankly, multiple decades of exploration of different types of different viruses to try to do this with. And over the course of the last 10 years in particular, one subset of viral vectors known as adeno-associated viruses have come to play an important role in being just that, being delivery vehicles for directing and providing back an extra copy of normal instructions for how to do the right biological activity. Spark's work led to what was the first approved gene therapy for a genetic disease in the United States just 5 years ago. Since then, there's been a couple of others gene therapies that have also been approved, and now has moved beyond diseases that Spark was working on, one of which was a rare eye condition where we would inject this viral delivery system directly into the eye, to now injecting it into the circulation, applications in neurodegenerative neuromuscular conditions like spinal muscular atrophy, and in blood clotting disorders like in hemophilia. And so, a tremendous amount of progress has been made, and we now have approved medicines based on this. And these are medicines that they hold the promise, because basically what you're doing is, instead of sort of correcting a biological process transiently, you're delivering the instructions so that the cell can frankly do the last step of manufacturing, if you will, itself. And therefore, once you give those instructions, it can allow it to do it repeatedly. And so we only actually have to deliver that instruction once. And that's really one of the in addition to fundamentally addressing the disease at its root cause, there also is the potential that you can only have to do this one time to really correct the underlying basis of disease. And so we've got examples of changing and restoring vision in patients who are blind. We've got examples of taking patients who have multiple episodes of bleeding over the course of weeks and years to now not seeing that anymore. Kids who are infants that suffer from a fatal neurodegenerative condition or neuromuscular condition now being able to sit up and walk and have normal developmental milestones. And these are things that have already happened and are already happening, and sort of begins and sets the stage for much of what Geoffrey mentioned as what we look forward to as the future.
Fascinating. Rahul.
Yeah, well, I don't know about you, but I'm very inspired by what Jeff and Jeff have just told us. We are actually sitting here living in the golden era of biology. We are actually talking about curative therapies. What Jeff and Jeff are talking about are things that can actually, with one shot, with one treatment, they can actually fix you. And that's an amazing world that we are going to be living in, and our children will be living in. And I'm super excited to enter that world, but I also want to be able to make sure that we are able to make these type of medicines available to as many people as possible. And what is something that we've recognized in the last couple of years is that while the biology is explosively growing and giving rise to these engineered therapies, and the underlying mechanisms of disease are being unraveled every day, we are not keeping up with the approaches to make these type of medicines for large populations, because they're made with like laboratory processes still. And these are different than pills and capsules that you guys are familiar with. These are viruses, cells, and they're not that straightforward. And so somebody's got to think about the technologies that you need to make these at low cost, at scale, so that large populations, as many people as possible, can get benefit from this. And that is the purpose of the company that we are running, called Resilience. And this company was formed 2 years ago in the middle of the pandemic. And all we think about 24/7 is how do we take advantage of this incredible, inspiring story that is unraveling in front of us and make these type of medicines available to as many people as possible. And I'll talk more about that as we go.
Geoffrey, anything you want to add?
I'll pick up on things that Jeff and Rahul just said, and that Jeff described that this allows you to go after the root cause of disease. And in fact, through all of the heroics of medicine, much of medicine as we know it today is really operating around the margins of treating symptoms, hoping to encourage progression-free survival, hoping to add increments to the baseline of today. And the ability to be able to intervene at the level of DNA, either of cells outside the body that go in, or of DNA of cells that are inside your body, it really for the first time allows one to start with the default expectation that you won't get to be a medicine unless you can cure a disease. So what we're doing in Tessera is pioneering a technology we call gene writing. And simplistically, what it allows us to do is to write short or long messages of DNA into the genome of human cells. And the reason that has the potential to be a big deal is all of us are different. That's driven by, in large part, the basis of the DNA that we're born with. That DNA is 3 billion letters of code. And unfortunately, if one of those letters is erroneous in a whole bunch of different locations in the genome, and that child is born tomorrow, they probably don't get to see their first birthday. And yet, if we could go to the exact site where that error occurred and change just that one letter, so scroll through 3 billion lines and change one, that child's life would change forever. I mean, it would be so transformational that the parents may not remember the name of the disease that the child was born with. And there's hundreds of millions of people around the world that have been born with that unfortunate kind of mistake that either predisposes them to a shorter life or daily exposure to disease. All of us, over the past 3 years have experienced the frailty that comes with fearing disease. I mean, this is a lifetime of disease. And we started the explorations that became Tessera about 7 years ago. This is as the first companies pioneering a catchy acronym called CRISPR were going public. And CRISPR is a sensational technology. For those of you that have heard of it, it is a programmable means by which you can go to a location in the genome and then cut the genome at that spot. Cutting the genome is something that our cells don't like, and it typically turns off the gene where that break took place. So that creates some value in medicine. Like, imagine when things break in your house, sometimes your toaster's on fire and unplugging it is a great solution. But most of the time, the diseases that Jeff was describing and that I was just describing, unplugging the thing that just broke doesn't improve the situation, in that when things break, they typically stop functioning. It's true in our household, it's true in biology too. So to fix that much bigger category of diseases, you have to be able to write, meaning to programmably write a short or a long sequence into a given location to be able to correct a genetic disorder or to add new function to cells. And the sensation of CRISPR isn't very good at writing, but that's not Mother Nature's fault. She invented it to be a great pair of scissors. So we started the company on the basis of having asked a couple years prior whether Mother Nature might have invented machinery that spent billions of years learning how to write DNA. And we quickly realized that we were thinking too little of her to even ask the question. The category that does that are called mobile genetic elements. And they happen to be the most abundant sequences in all of life as we know it. So about half of every one of our genomes was built by these sequences of DNA that learned how to write themselves into new locations. About half of the genome of any pets you have at home, animal kingdom, plant kingdom, and more. So if you think of the abundance of a sequence of DNA as a proxy for how much lab space Mother Nature gave that pet project, she gave more of the lab over the last 3 billion years to mobile genetic elements than anything else. So the question went from, do these exist, to, oh my God, like where would we start? Which of these still work? Which could work on the human genome? Which could we program so that they're going to a desired location and inserting a therapeutic sequence, not their own code? And even better, which can we program both code that goes in and location for inserting code? And mostly as a testament to Mother Nature, we've made some amazing progress. So we have gene writers that can change any one base pair to another. So A to a T, A to a G, A to a C, and vice versa. They can make small programmable insertions or deletions. Like that category by itself is applicable to about 90% of human genetic disease. And then we've sort of doubled down on machinery that can write very long sequences, entire genes into given locations. And in that realm, we've done something that, as far as we know, is a first in the history of molecular biology, which is give just 2 RNAs. So imagine a therapeutic composition akin to the SARS-CoV-2 vaccines, lipid nanoparticle RNAs inside. But in this case, the RNAs that we deliver are able to go into a cell and letter by letter write one of those RNAs into a sequence of DNA that stays in the genome of those cells as long as they live. And that allows one to contemplate being able to correct genetic disorders across a wide variety of cell types. And we're also doing things like engineering T cells inside the body with a redosable lipid nanoparticle so that they can go on the hunt for cancer cells without ever having to take them outside the body of the patient.
Fascinating. Okay, so let's do a deeper dive into gene cell therapies and then where do we think the field is going. I'll start with you, Jeff. Like, most of the current programs in gene therapy are actually in diseases that have a clear genetic cause of the disease. Where do you think the field is going? When is it translating to common conditions? And what are the areas that we should focus on? And what are the areas that investors should focus on? What's the time horizon to the clinic?
Well,
a couple of thoughts on this. So first of all, it's important to kind of keep in mind that there are about 7,000 rare conditions, of which, somewhere the last time I checked, it's maybe in the order of a couple hundred had some existing treatment. And when I say treatment, most of those, as Geoffrey described, are things that are symptom treatments, palliative in some way, but they're not necessarily getting at the root cause of disease. So 7,000 of these rare conditions that affect collectively about 30 million people in the United States, about 10% of the entire population, and that generally extrapolates globally. So it's actually a pretty large space still to do a lot of work on, and to do, first and foremost, a tremendous amount of good for patients who are waking up every day and dealing with the fact that that disease is confronting them and a part of their day. The second thing I would say, though, is there's a tremendous amount that we often and consistently learn whenever we work in rare conditions on things that are applicable to non-rare conditions, to complex disorders, because perhaps what we're trying to do is to figure out how to deliver some of the exciting stuff that Geoffrey is talking about, but we haven't yet figured out how to do it to this particular organ. And yet, if we could do that, and when we do that for a rare condition, it makes us now realize, well, we figured out the delivery, now we could work on changing and using a different tool out of Geoffrey's toolkit, company's toolkit, to work on a complex disorder. So I think there's tremendous learning still to be made there, and I think it's still a lot of progress and a lot of diseases that are there still to solve with the current technologies applied to rare conditions. I would also just, to piggyback on what Rahul said, there's also there's a huge amount of opportunity when you think about these particular technologies outside the United States, and then outside in particular the what we would consider the developed world, where frankly, when you think about the potential to do a one and done treatment, the complexity and the lack thereof of infrastructure in many of the world's countries where they do not have the infrastructure to bring, say, someone to a clinic daily, or to be able to get them a medicine that needs to be frozen at -80 degrees. The fact is that these treatments that are one and done, where you could bring someone from a rural area in a country to a facility once, opens up a huge opportunity. But we can't get to that without sort of scaling it the right way, without investing in the technology to be able to make this the right way and drive costs down. And so I do think that is another area. I'd say if you were to talk thematically about what are the two sort of themes, I would say even more so than jumping from rare conditions to complex conditions yet, is the need to continue to build out a scalable, cost-effective, consistent platform for manufacturing many of these different types of technologies. And secondly, I would say also bringing more innovation to the development of these medicines and conducting clinical trials, which is also something that, much like manufacturing, I think has the ability to make substantial progress come from where we've been today.
And I think that's a great introduction to our next question, Rahul, for you, around the manufacturing, right? Cell and gene therapies are evolving, but there's high cost of manufacturing. It's a highly skilled area. Because it's developing in individual small biotechs, there's often even lack of processes standardization. How are you dealing with those challenges at Resilience, and how are you thinking to allow us to remain the world leader in biotech and really enable these technologies to reach the patients?
Yeah. So I think the first thing is that the industry, the way it is set up right now, everybody that is trying to develop these drugs are also trying to figure out how to create the technologies to make these drugs. And that kind of approach was okay 30, 40 years ago when drugs were coming out like once in a blue moon through the discovery process of chemical libraries. But now, as Jeff is saying, you've engineered things, and so there is an avalanche of new drugs through these platforms that they're developing, which once you figure out that platform, you can have like a lot of drugs that can come from that. And what Jeff was saying, like you're going from like rare to more complex diseases, more people are affected, more common diseases, so more people need them. And so and the third thing is that these drugs also shrinking the timeline for development, because the results are so black and white. When you give these drugs, it's not like an incremental improvement, like it's like you cured somebody. So the regulatory authorities are saying, like, guys, the patients are waiting, make these available as quickly as possible. All of these things are making it very, very difficult for companies that are product developers to also do technology development. So the time has come in this industry to move away from this cottage industry that we propagated for the last 30 years as we build this industry. We have to say, like, guys, look at other industries that are giving incredible consumer value to us. Look at electronics, chip making. Does every company that gives you a product make their own chips? Does Apple make its own chips? Does Qualcomm make its own chips? Does AWS make its own chips? Does NVIDIA make? No. They depend on one athlete or two athletes at most that are best in its class, because that's all they do. Taiwan Semiconductor is an example. In our industry, everybody wants to do everything. Well, the gig is up, guys. Patients are waiting. And if the pandemic was a catalyst, so be it. Because that pandemic actually did tell you, like, you got to stop this insanity. You need to give this work to the people who really do this all the time, rather than diffuse your mind space doing 30 different things when you're only good in one. It's an age-old economic principle of division of labor through comparative advantage. We've forgotten that in our industry. And we need to stop. And that's what this company is all about. That's what we're trying to do. We are focusing our scientists and engineers to work on the technologies that will allow the cell and gene therapies to be scaled, to think differently, to not go back to 30-year-old papers that an academic wrote and put it in a factory, which is what our people are doing right now.
Yeah.
So I'm not going to go into the gory details of the specific technologies, but all I can tell you is the concept that our people are not thinking about animal experiments. We're not thinking about doing clinical trials. We're not selling drugs. All we do is think about how to make these drugs better. And that's the answer from my standpoint. That is going to be the transformation that this industry needs so our children can live in a better world.
Absolutely. If the new inventions cannot scale and be brought to patients, we don't have an industry, and we technically failed. And certainly, I guess like even in our own career at Gameto, we were saying that if you want to be an expert in reproductive medicine, you cannot be an expert in cell engineering and in manufacturing at scale. And it's absolutely essential what you're doing and important. That's why you got the backing from the US government, so that we really are able to remain at the forefront of biotechnology innovation. And then I guess I'll switch to cell therapies, which is kind of like turning science fiction into real science, right? Since Yamanaka won a Nobel Prize in 2012, you can now make a stem cell in the lab, you can engineer that cell to become a mature cell type, and you can cure disease that is caused by that cell damage. And we've seen some early take-up in cancer, in blood disorders, now we're seeing it in diabetes. What do you think about where the translational going in cell therapy, Geoffrey? And I know you obviously co-founded Sana that is working on cell engineering. Could you tell us a bit more about that?
Yeah, I've been tempted to jump in on a bunch of the great comments that my colleagues have made, and this will connect to the premise of, are these just applicable to rare diseases or the diseases that all of us experience at some point in our lives, or in our immediate family and friend circles. In that the basis of Sana was twofold. One, the ability to engineer small vesicles that come off of cells so that they can programmably enter cells in your body and deliver a genetic payload or another. The second was to be able to engineer the surface of cells themselves so that Dina's cells could be injected into my body, and as opposed to being voraciously rejected, which is what happens with everything outside of red blood cells, my immune system would recognize them either as self or would be unable to recognize them as foreign. And if you can do that, i.e. hide a cell from your immune system, you open up a huge number of doors whereby you can start to think about the kind of manufacturing that Rahul described, delivering not just kilograms or tons of a small molecule, which has been most of the last 100 years of medicine, or the same for a protein therapeutic, which has been the mainstay of biotechnology for the past 30 years, but delivering a living, stabilized cell that could be injected at a primary care location or at any location into a patient that can benefit from that cell therapy. So the insight that came out of the lab of another of our co-founders was that if you knock out a whole bunch of stuff that reveal that a cell came from another person, and you overexpress a protein that is called the don't eat me signal, which actually triggers immune cells that are about to engage and engulf and destroy a cell to disengage. It's sort of like the Obi-Wan Kenobi, these are not the drones, the droids you're looking for. Then you can have remarkable abilities to be able to take every single cell type that we have tested and hide them from the immune system of a recipient. So what we've done so far is show that cells of the immune system, including T cells, if brought into a non-human primate, either derived from human or another non-human primate, can be hidden from the immune system. Same for the beta cells in your pancreas that produce all of the insulin in your body. Same for cardiomyocytes, the muscle cells that allow your heart to be able to beat. Same for neuronal cells and a few others. So to take just the diabetes portion of that, it's about 3 million people in the Western world that have type 1 diabetes. There's a much bigger number of people that have type 2 diabetes. With a population of cells the size of like a small marble, like smaller than a dime, you could inject into a number of different locations in the body and create an artificial pancreas that would, if hidden from the immune system, provide them the ability to have normal insulin regulation and thereby be free of the glucometer, the frequent injections of insulin doses, and the corresponding quality of life and quantity of life implications of being a patient with type 1 diabetes. So that is among these tipping points where we don't know yet if we're right. We're going to be bringing this into the clinic. But if we're right, this is going to be a really valuable new category of medicines. And per the avalanche comment that Rahul made, one of the interesting aspects of this era of medicine is akin to Moderna's success. Stefan at Moderna had famously always said that Moderna could never have one drug. It was either going to be zero, or if successful in any one location, it was going to be a whole bunch. And that is true for many of these platforms. Like, it's virtually impossible for Tessera to be able to address only one genetic disease. Like, it's either going to be zero, or it's going to be a whole bunch. And same for these kinds of lessons where you can hide a cell from the immune system, or comparable advances taking place across the field.
Fascinating. I like the diabetes example because that is something that we're already seeing in human trials, and it's literally fascinating. You tell somebody a story and they're like, wait, this is real? This exists? Rahul, maybe a question for you. You recently announced with Resilience a partnership with California Institute for Regenerative Medicine. How are you thinking about cell therapies, any areas of priority?
So cell therapy is a very broad category, and the interactions that we have with California Institute of Regenerative Medicine is based on one particular type of cell therapy, which is based on making these type of products that Geoffrey was talking about, which is these artificial pancreas in this case, or other type of products. The kinds of technologies that we are going to be mastering in terms of making is like, you can actually take any cell from your body and convert them into a primordial cell called a stem cell, and then have a mechanism to take it to any particular type of tissue that you want. This approach to, on demand, produce cells that you need, whether it's neuronal cells, whether it is pancreatic islet cells, whether it is liver cells, it increases the plethora of potential curative therapies way beyond transplantation, for example. You don't have to wait for a liver transplant if you have this kind of an approach. So what we're doing with CIRM, California Institute of Regenerative Medicine, is to work with the companies that are actually working on these type of amazing ideas that are funded by the California Institute of Regenerative Medicine, and then becoming a manufacturing partner for them so that we can provide them that support that they need to be able to translate their innovation, the biological innovation, faster to patients. But we're not just doing it with regenerative medicine. We're also working on immunotherapy, which is the other arm of cell therapy, which is very important, which is take cells that can be engineered and put back so that immune system can fight against things like cancer. And so these, I'm sure you know about CAR T type of cells that are, 5 of them are in the market now, and they do amazing things. I mean, everything fails, and these things can go on and have amazing patient results. But again, the production of methodologies of these type of amazing therapies is still very young and not scalable. And somebody's got to think about how to do this. And that's something we are doing. And we're also working with the Barker Institute of Cancer Immunotherapy, for example, which is the largest network of innovators in the United States that work on cancer immunotherapy. And again, they are more interested in the biological innovation, the clinical trials, et cetera, but we come in and help them out with, or the idea is to come and help them on the manufacturing so that, again, patient impact is accelerated. So that's the kind of work that we're doing on cell therapy. But the bigger point is that, again, decoupling product development with technology development and applying focused efforts on coming up with better technologies that can actually have meaningful impact on cost of goods and scalability so that more people can have access to these amazing medicines at affordable prices is the mission of the company.
Okay, so it seems to me that you've cured genetic disease, you're solving manufacturing, you're writing genes and writing cells. It appears that the pace of this biotech innovation has never been faster, but the valuations of both public and private companies have never been lower. So how will this, as I guess a final discussion topic for all my panelists today, how will this play out? What are you thinking about biotech in the next 5 years? And are there any other technologies that you think we should be aware of that will make a difference even beyond some of the ones that we've discussed, like cell and gene therapy? Let's start with you, Jeff.
Yeah, I mean, first of all, I would say that this is, I think it's always important to zoom out. And so when you talk about multiple quarters of valuations that are different than maybe they might have been a year or so ago, I think it's important to zoom out because the thing that will drive progress is the continued acceleration of more scientific discovery, whether they be tools that allow us to engineer, genetic engineering or cellular engineering, or whether it's also advances in understanding of the biology of specific disease. And that's, as critical, by the way, as the ability to engineer. And so I think those are going to drive fundamentally more progress. I will say that I think it's a period that is, in my view, generally a healthy one, where we can actually look critically at the fact that there are probably multiple companies, many companies in different categories, which may have started based on an idea, but which, frankly, you probably could have done with a smaller number of those companies, or you could have done with a company focused on one particular application. I mean, my general observation, when you have this number of new platforms that are being developed at the pace they're being developed, whether they're gene writing, whether they're gene editing, whether they're base editing, whether they're prime editing, whether they're epigenetic editing, is that they all have to find a place where they are the best tool to fix a particular disease. And so I think one of the things that you will see come out of this period is a better understanding and match between the technologies that are proliferating as they are and the disease biology and what's the best tool to use in that application. And so that rational view and understanding of sort of where you're trying to take the tools, I think is what will come out of this, as well as the fact that you'll have opportunities to basically find a particular medicine that might be being developed in one company, but frankly probably should be moved over into another company, or those should be combined. Those are things I think that are presented by this opportunity. But that's all about corporate development and financing considerations. The fundamentals are not changing. The fundamentals are actually changing, if they are, more positively and rapidly. And what I think you will see is the formation of better, stronger, more capable companies that will drive more medicines to patients and be more selective about how and where they match up the tools and technologies that we have with the diseases that they can go after, and perhaps with the type of relationships with organizations like Rahul, where they figure out what they're really great at, and they do that extraordinarily well. And then they have partnerships to be able to make, ultimately, what we're trying to do is to get a medicine to a patient.
Absolutely. Rahul, any comments on that?
Yeah. So firstly, I think this industry hasn't seen like an increasing interest rate environment, I don't think, in a long time. So this is a very sort of a new time, a new twist, I would say. And I think whenever you have higher or an environment of increasing interest rates, I think there is a more and more emphasis is given on earlier revenues. And so, biotech by definition is something where, the ships are coming, 10 years later loaded with gold, but, we need something now. And so obviously valuation is going to go down in this environment. And the approach that I see, how the industry will react to something like this is that I think people will have to become much more capital efficient. And this kind of goes back to again, don't try to do everything and piss away a bunch of money and make preventable mistakes. Try to work with people that are good in what they do, and try to conserve your capital, your mind space, your energy to what you are excellent in, and drive forward the innovation as quickly as possible, closer to commercialization, closer to patients, without burning up a bunch of capital. So that is one thing that I see happening in the next several years in terms of how this industry is going to change. The other thing that I see is that I think that the pandemic showed us that you cannot rely on supply chains that are based on stuff coming from different countries, and pandemics or other types of things can really create major issues. And so I see a lot of resiliency of supply chains to come from local production and local reliance. So there's going to be a lot more emphasis by governments to control their supply chains rather than rely on other countries where geopolitical issues can screw up things. So I think that vaccines and pandemics and infectious diseases, population-based issues are obviously the top of mind issues, but that's also going to create more and more emphasis on other type of drugs where there's inequity between populations. And so governments have a purpose to try to make this different, and it's kind of going to a little bit reverse of globalization to become a little bit more control of supply chains. So that is something that we're looking at. I mean, of course, you all know about the executive order that the President Biden actually issued yesterday, which is all about emphasizing investment in this country and bioeconomy and biomanufacturing, as speaking today in Boston. So we are watching that very carefully and of course trying to be part of that solution for this country.
Absolutely. Biotech is going to be very important for us. Defense industry, and we'll talk about it tomorrow with Bob Nelson, your partner. Geoffrey, give us your take. We're hearing that the current markets are going to exert evolutionary pressure for companies to find, let's say, what we call product market fit, be more collaborative. What's your take?
So each of the companies I'm a co-founder of, I'm an inventor of the founding technologies. I spent all my time thinking about technology. So I don't have any sophisticated market views. But I do observe from time to time that although everybody knows buy low, sell high, it is amazing how often individual human tendency and herd tendencies are the exact opposite. Like if you had observed the degree to which the biotech pullback was the steepest in the history of all of biotech a couple of months ago, and found places where there was a really high quality company in which that was being overlaid unfairly, you would have received a remarkable return in just the past 3-4 months. And I'll jump back to kind of the longer picture, which is, biotech and other fields go through these bubbles or sensations or kind of fits of hopes that it's going to happen all at once. And then it's easy to get desensitized by that and miss the point in time when it really happens, or the period of time over which it happens. And with certainty, I mean, it is hard to know much about the future, and the pandemic has shown us that, but with certainty, the transition of biology from being a really expensive game of chance, where a couple % of new drug applications or investigative drug applications actually become successful therapeutics, to one where 90+% of them become successful therapeutics, it is going to create trillions of dollars of either rearranged therapeutic value or completely new therapeutic value. I mean, that is going to happen on the next 10+ years. And, it's going to be a subset of companies that are able to base all of their decisions around their particular version of that future and get the components that Jeff and Rahul described right. I mean, the explosion of possibilities in biology is going to lead to more and more roads to Rome, where there's like another way of potentially treating a disease. But there's going to be a couple highways. Like, in Tessera, we describe the future as having what we think of as win state therapies. Win state meaning a therapy that is so good that it will not obviously be displaced by anything. And in fact, I think unless you can conjure up a perspective of what that medicine will be, and with confidence aim your technology towards that profile, one risks creating transient value versus enduring. I mean, we've gone from an era where it's been almost impossible to come up with any new medicine, let alone any new type of medicine, like new category, to where the number of distinct modalities, technologies being poured into human disease is expanding rapidly. And it's not going to make it simple, but the companies that win over this next era are going to create extraordinary impact in the world, and it's going to be really exciting.
All right. Well, with that, we hope that we convince you that we're in the golden age of biotech. And thank you very much for a lively discussion, for coming today.
Thank you.
Thank you all.